Thank you for the introduction. I'm so glad to be here today to get to talk about one of the projects that I have been working on for the longest, which is looking at studying bat pollinators in southern Thailand. So most of us, when we hear the word pollinator, we probably think of bees or butterflies or other insects. And that makes sense because these are the pollinators that most of us probably see the most often. These are 3 video clips that I took here in Thailand. On the far left, we have Apis dorsata, or the giant honeybee. In the middle, we have Apis florea, or the dwarf honeybee. And on the right, we have a glassy tiger butterfly. So not only are these the species of pollinator that we see the most often, they're probably also the ones that we hear about the most often in the news as well. Usually when the news talks about pollinators, they're talking about honeybees or other bees in general. But in addition to these diurnal pollinators, there are some really important nocturnal pollinators as well, including moths and bats. So here are 2 other video clips that I've taken here in Thailand. And these show nectar bats that are foraging on the nectar of flowers that bloom at night that are bat pollinated. And we generally don't think about bats as pollinators because we don't see them usually. Usually at night when they're foraging, we're in our houses. And even if we leave our house at night, we probably wouldn't think to look for them. And if we did try and find them, 1, the flowers that they pollinate are really tall, way above our heads. And 2, since it's dark outside, we would have a hard time actually seeing them unless we had like special equipment like this infrared camera that I use to take these videos. So since most of you probably don't know much about bat pollinators or bats in general, I wanted to give a brief introduction about bats in the Old World Tropics. So the Old World Tropics includes tropical areas of Asia, Australia, and Africa. So that includes Thailand.
Asia, Australia, และแอฟริกา เขตนี้มีค้างคาววงหนึ่งที่เรียกว่า Terrapoded,
such as these images shown here. But there is one family in the Old World Tropics called the Terrapoded, and this family consists only of fruit and nectar feeding bats. And so here are some examples of Terrapoded bats. So there are a number of key differences between these insect-eating bats and the Terrapoded bats. You probably noticed that the insect-eating bats tend to have relatively large ears and relatively small eyes, whereas the fruit and nectar-eating bats have relatively small ears and relatively large eyes. And the reason for that is that the insect-eating bats, like almost all bat species worldwide, rely on echolocation to navigate and forage in the dark. So they don't really rely on vision at all, so they have really poor eyesight, but they have these large sensitive ears that help them with echolocation and navigating in the dark. In contrast, the Terrapoded are the only bats in the world that are not capable of true echolocation. So they never evolved large, complex ears because they don't rely on them. And instead, they rely on vision and scent to navigate and find food in dark environments. So they have really good eyesight under dim conditions. Now, within the Terrapoded, there are some species that are strictly nectar bats. So they only consume nectar from flowers. And these species tend to have really long snouts and long tongues that help them get to the nectar and flowers. Then there are other species within the Terrapoded that are primarily frugivorous, but they will also occasionally visit flowers. So this is an example of a mainly fruit-eating bat that has a much shorter snout, short tongue, but broad, powerful jaws that help it to bite down on fruits. Within the Old World Tropics, in addition to these bats, there are also a number of plant species that are bat pollinated. You may recognize some of them, such as this banana flower, or you may recognize the durian fruit. I'm not sure how many of you have ever seen the durian flower before, but here it is. And then other species, you may not recognize the flower, but you may be familiar with the name. So from left to right, their names in Thai are Nun, Turian, Gui, Peka, Sata, and Lampu Lampan. So how do we know that these species are bat pollinated? Well, first of all, they all bloom at night. So these species typically start blooming in the late afternoon or early evening. They reach peak bloom at around dusk, and then the flowers only last for one night, and then they fall by morning. So depending on the species, some flowers will fall as early as 2 a.m., others fall around 6, 7, 8 a.m., but their peak blooming is during the night. So we know that they're pollinated by nocturnal pollinators. Another way that we know is based on the flora morphology. They have traits that are attractive to bat pollinators. So all of these flowers tend to be either white or cream or yellow, because these are the colors that are easiest to see at night. Even banana flowers that have this red bract, the actual flowers themselves are white and yellow. And remember that these bats are relying on vision to find flowers. So having a light or pale colored flower makes it easier for the bats to find the flowers. These flowers also typically have a relatively strong scent, and that also helps the bats find the flowers in the dark. Other features include the fact that these flowers are all relatively large because they have to be able to withstand a large bat landing on them, and they produce lots of nectar and pollen because bats eat a lot more nectar than bees would. So another way that we can verify that these plants are bat pollinated is by using an infrared camera and monitoring the flowers. Is there a way to erase this annotation right here? Okay, I think I figured it out. Okay, so in this video, let me go back a minute. Okay, so in this video, we see some bats visiting a Parkia flower or Sata flower. And this is another way to verify that these flowers are visited by bats. So we can see the bat land. It kind of moves around and licks the nectar from all sides and then flies away. But the thing about this is that we know that there are often insects that visit the flowers as well, because nectar eating insects, they like the nectar. And so, for example, this Parkia flower has lots of nectar. So insects will come and drink the nectar as well. And so that leads to the question, who is actually pollinating these flowers? Is it the bats or is it insects? And to answer that question, we can use a pollination experiment. So in a pollination experiment, we leave some of the flowers unmanipulated, and this allows all animals to visit the flowers as normal. So that includes bats and any insects that want to visit. This is our open pollination treatment. Some of the flowers we enclose in a cage, and the holes of the cage are large enough that insects can still go in and visit the flower as normal. But the bats are too large, they can't get inside the cage. So we know that bats are not able to pollinate these flowers. So these are our insect pollinated flowers. And then some of the flowers we can enclose in a mesh bag, and this prevents all animals from visiting. And this is to confirm whether pollinators are actually needed, because some species don't actually need pollinators and they can set fruit without them. So a previous study by Dr. Bumrung Z from Prince of Songkhla University found that Parkia relies on bats for pollination. And in the study, they studied 2 species of Parkia. The Thai names are Satha and Riang. And in this first treatment, the open pollination treatment, they found that pollination success was around 70%. That means that around 70% of the flowers developed into fruits after being visited by all animals, bats and insects. In the second treatment, where animals were excluded, 0% of the flowers developed into fruits. And in the insect pollinated treatment, some of the flowers did develop into fruits, but the median was still at 0%. So the insects don't really contribute much to pollination, which means that the bats are doing pretty much all of the pollination for both species of Parkia. A similar study with durian found the same results, where flowers that were exposed to all visitors were able to set fruit. We see here around 10% of flowers developed into fruits. But for flowers that were in the animals excluded treatment or in the insect pollination treatment had fruit set of 0%. So for those of you who like eating durian, now whenever you eat durian, hopefully you'll remember to think of the pollinators, the bat pollinators who allow us to eat durian. And then this work was done by a master's student who was studying 2 species of Soneratia mangroves for her thesis. Soneratia ovata is classified as near threatened, and Soneratia griffithii is classified as critically endangered. So for her thesis, she was looking at the pollination ecology, and from her results, we see that this is the animals excluded treatment for the 2 species. Fruit set still occurred, but it was much lower compared to the treatments where bats and insects were allowed to visit. And for ovata particularly, while the insect pollinated treatment set fruit as well, it was even higher when bats were allowed to visit. So these data show us that while these species are self-compatible and don't necessarily need pollinators to produce fruit, they have much higher reproductive success when they are visited by pollinators, both bats and insects. So one of the disadvantages of a pollination experiment is that we don't know which bat species are visiting the flowers. We just know, okay, bats pollinate. But there are many species of bat, nectar bats in Thailand that visit flowers. And this slide shows 7 of the most common species in southern Thailand that visit flowers. The 3 on the left are nectar bats that only visit flowers, so they never eat fruit. And then the 4 species on the right are primarily frugivorous, but also occasionally visit flowers. So you're going to be hearing me talk a lot about these 7 species. So I want everyone to take a mental picture of them. Remember just in general what they kind of look like. And now, how many of you can identify the bat species that are visiting these Aroxylum flowers or these Peka flowers? It's very difficult to do because, like I said before, not only are the flowers 5, 10, 15 meters up in the air, but it's dark outside. And even with our infrared cameras, the resolution isn't that great, and the bats are so quick that it's really difficult to identify them to species. So if we want to know which species are pollinating the flowers, we have to rely on using mist nets. So this photo in the middle shows a mist net that we used in front of some Peka flowers, some Aroxylum flowers. And during the day, we can see the net. It's kind of difficult to see it because the lines are so thin, but it's visible. But at nighttime, when it's dark, this net is basically invisible. And so any bats that come foraging to these flowers end up getting caught in the net, and then we can lower the nets and untangle them and identify them to species. So when we catch the bats, there are 2 main types of data that we're interested in. The first are visitation rates. So for each bat species, how many bats are we catching per hour? And the second is collecting pollen loads from them. This is a bat that we caught that was carrying a lot of pollen. Usually we aren't able to see the pollen with the naked eye. You know, we would usually have to use a microscope. This one was carrying a lot. But for every bat that we catch, we use a special gel and a syringe, and we run the gel through the bat's fur. And the gel is kind of sticky, so it picks up any pollen grains in the fur. We mount it on a slide, and then we can use a microscope to identify the pollen grains. So in this photo, we can see a banana pollen grain, a durian pollen grain, and a Parkia pollen grain. And this allows us to identify the flower species that this bat has been foraging at. So we use these two types of information in order to calculate pollinator importance. Pollinator importance is simply the product of visitation rate times pollen load, because bat species that visit but aren't carrying pollen aren't going to be able to pollinate the flowers, compared to a bat species that not only visits often, but is also carrying pollen of that flower species.
ค่ะ และสำหรับอัตราการถ่ายเรนูแม้สัตว์บางชนิดมีอัตราการเยือกเย็นสูง
The larger the number, the more important the bat species. And on the x-axis, we have the 7 most common bat species that I mentioned earlier. So this is the data for Ceiba Newton. This is the data for Peka or Araxilum, and this is the data for Sata or Parkia. And what we see for these 3 species is that there is only one bat species that is pollinating the flowers, and that is this bat ES right here, which stands for Eonycteris spalaei, which is this species right here. So these 3 species rely on a single bat pollinator. But other plant species have multiple pollinators. So for example, the durian flowers are visited by both Eonycteris spalaei and also Rosettis fleschenalti, which is this species right here. For banana flowers, the most important pollinator is Macroglossus sabrinus, which is this species. And for Sonoratia mangroves, the most important bat pollinator is Macroglossus minimus, which is this species right here. So we can see that there's some variation. Different plant species may rely on different species of bat to pollinate them. But in general, Eonycteris spalaei is like the star pollinator of many of these night blooming species. Here are some photos taken by the talented Merlin Tuttle, these are all photos of Eonycteris spalaei, or the cave nectar bat. And you can see that it likes all different types of flowers and does a good job of collecting pollen from them as well. So because Eonycteris spalaei is visiting all these different types of flowers, it led us to wonder, are they transferring pollen across different species, which would be bad for the plant, or are they mostly transferring pollen between flowers of the same species? And we hypothesized that because these flowers have different morphologies, different appearances, that they would place pollen on different areas of the bat's body, and that would allow them to move pollen between flowers of the same species. So in order to test our hypothesis, we conducted a flight cage experiment, and in each experiment, we used a male flower and a female flower. So in this case, in this trial, we have 2 banana flowers. Right now, the bat is foraging on the male flower, and we keep the female flower covered initially because we want it to feed on the male flower first. So in a minute, when this bat is done, we're going to remove the bag from the female flower, and then wait for the bat to come back to the female flower. and let it feed on the female flowers. And then we can count the number of pollen grains that the bat moved from the male flower to the female flower. And for some of these trials, we used 2 flowers of the same species. And for other trials, we used 2 flowers of different species to see how much pollen they're moving between flowers of the same species versus different species. And what we found was that they move more pollen between flowers of the same species. So the green dots indicate combinations where we use the same flower species, and the black dots indicate combinations where we used different flowers of different plant species. So for example, for ceiba flowers, for Docknuden, we can see that when they're given male and female ceiba flowers, they transfer around 200 pollen grains. When they're given 2 durian flowers, they transfer around 50 pollen grains. When they're given 2 banana flowers, they transfer around 100 pollen grains. But when they're given combinations of different flower species, they transfer no more than 30 pollen grains on average. And the reason that this happens is because, as we hypothesized, the flowers place pollen on different areas of the bat. So when the bat lands on the banana flower and puts its nose in the florets, this is where most of the pollen gets deposited on the face. For Parkia flowers, it kind of ends up hugging the flower, and so most of the pollen gets deposited on the bat's chest. And then for 2 other species that we examined, ceiba and durio, these flowers belong to the same family. They have similar floral morphologies. So the pattern of pollen deposition is very similar for the 2 of them, but the bats never transfer pollen between these 2 types of flowers because they flower during different times of the year. So ceiba flowers from December through February mainly, and durian mostly flowers in March and April in southern Thailand. So given that we know Eonectarus is visiting these different species of flower, we also wanted to look at its diet throughout the year. And these graphs show plant phenology in the colored bars. So that refers to how many individuals were producing flowers. So for example, for ceiba, we can see that for most of the year, the trees don't produce flowers. And then around November, December, January, February, they produce a lot of flowers. And then the dashed line represents the bat pollen load. So how many pollen grains of the species are the bats carrying? So when ceiba is flowering, the bats carry a lot of pollen. When there are no flowers, obviously they aren't carrying any pollen. Same thing with durian. The main flowering season is in March and April. So when these trees flower, the bats visit the flowers and carry a lot of pollen. During the rest of the year, they don't really carry much pollen. But for the 3 other species, these are individuals that produce flowers pretty much throughout the entire year. So there are always at least a few plants that have flowers. And we can see that Eonectarus spalaei also carries pollen pretty much throughout the entire year. So these 3 species in particular are important in the diet of Eonectarus because it provides food for them throughout the year. But when ceiba and durio are flowering, then Eonectarus really likes these flowers as well. Now, I'm sure based on the title, a lot of you are wondering, okay, I've been collecting data for 10 years. Have I noticed any differences over the past decade other than the fact that I am now 10 years older? And I want to start by showing you data that I collected during my PhD. So I have data from 2011, 2013, 2014 for 4 plant species. This is Musa or banana, Araxylum, Pega, Parkia, Sathaw, and Soneratia, Lampu, and Lampan. And the first thing that I want to bring your attention to is the fact that there are differences across plant species. So if we look at the banana flowers, there are many bat species that like to visit. So on the y-axis, we have visitation rate. So of the 7 main bat species, several of them like to visit banana flowers, compared to for Soneratia, we really only see 2 species that visit. And this makes sense because Soneratia is a mangrove plant, and most of the bats are not foraging in the mangroves other than these 2 species that are like these flowers in particular and are kind of specialist pollinators for Soneratia flowers. The other point that I want to bring your attention to is the fact that there is, even within the same plant species, there is variation across years. So for example, in 2011, we see relatively high visitation rates for many of the bat species, whereas in 2014, we really only caught 2 species of bat at banana flowers. Okay, now I'm going to add the data for 2019 and 2020, and it's a lot of data to look at once. So first, I want to bring your attention to only the cave nectar bat, or Eonectarus spalae, which, as I have mentioned before, is the main pollinator of many of these plants. So we can see that in general, visitation rates by this species has remained relatively constant across years. For banana flowers, we can see that the visitation rate is around 1 bat per hour. For Aroxylum or Peka flowers, we can see that there is around 1 bat every 1 or 2 hours. And then for Parkia and Soneratia flowers, we can see that there are around 1 to 2 bats per hour. So it appears that this cave nectar bat, the visitation rates have seemed to be fairly consistent over the past decade. Now, if we look at the 6 other species, I want to start by saying that I'm hesitant to make any broad conclusions, any conclusive statements at the moment, because this is like very new data that I just threw the graphs together for this presentation, and I haven't had a chance to analyze or do any of the statistics yet. So I don't want to make any conclusive statements about these other species, but from these graphs, it appears that visitation rates for many of them have decreased over the past decade. So if we look again at the banana flowers, we see relatively high visitation rates for many of the species in 2011, whereas in 2019, 2020, we see lower visitation rates. So I don't want to state that, oh, this indicates that the bats are in trouble or have declining populations. We don't know, or based on this very preliminary analysis or presentation of the data, it could be that they are simply using other food resources, or they may be moving into different habitat areas. So again, I don't want to say that, oh, it looks like these bat species are declining. This is very, very preliminary, and I also have data from 2021 that we haven't looked at yet as well. So maybe if APIA is giving talks next year, I might have some more conclusive data to share with you guys. But to wrap everything up, we know that bats are important pollinators for many night blooming species. Eonectarus spalae in particular is an important pollinator that pollinates agricultural crops such as durian and sathaw or parkia, as well as ecologically important species like wild banana plants and mangroves. And these bats are efficient pollinators due to the evolutionarily specialized relationship between the bats and the plants. So that allows them to to pollinate efficiently and that they mostly move pollen between flowers of the same plant species, and there's not much crossover between flowers of different species. And so the plants rely on different pollinator species. So even though Eonectaris spalae is a key pollinator for many of the plants, some of them rely on other bat species for pollination, which is really important in terms of conservation for both the bats and the plants. I want to go back again to the mangrove species that I said was critically endangered, Soneratia griffithii. And the main reason why this species is critically endangered is because as humans have developed the coastline, we've cut down a lot of the mangroves. But another reason is that the main pollinator of Soneratia griffithii is Macroglossus minimus. And this is a bat species that roosts and lives in the mangroves. So as the mangroves are being cut down, it's having a negative effect on the bat pollinators. And that in turn is going to have a negative effect on the reproductive success of these mangroves. So it's kind of a double whammy. Not only are they being cut down by humans, but they're also losing their pollinators. And then finally, in terms of long-term patterns, I would venture to say that Eonectaris spalae seems to be visiting relatively consistently. The other species, again, I want to go through and actually analyze the data before I give you guys any answers. But regardless, I think that this 10-year data set is going to give us some interesting answers about these important bat pollinators. So with that, I would like to thank everyone who's helped with this research, including colleagues from Mahidol University, Prince of Songkhla University, University of Maryland, all of the numerous students who have helped with data collection, and all of our funding sources. And thank you to the audience for your attention, and I would be happy to answer any questions that you guys have.